Hi, it's Paul. Let's have a look at the pathogens that you can see in respiratory specimens. They're many and varied. Most of these occur in opportunistic fashion, so they're seen in immunocompromised patients. Maybe one's undergoing chemotherapy or some underlying immunosuppression like HIV. First one we'll look at are the viruses, and these show typical viral cytopathic effects like we've looked at in the cervix. This is herpes, and you can see these are cells that's showing margination of chromatin. Over here we've got some moulding and multinucleation, and probably a large inclusion in that one as well. Here's some others in sputum with large eosinophilic inclusions, multinucleation. And you can see these could be a little worrying if you don't click to the fact that they have that kind of glassy marginated chromatin. Just another example there of them. CMV is seen here. We've got a BAL, I think, with macrophages, some bronchial epithelial cells, and then these cells which is showing an owl's eye appearance, so a really large cleared nucleus with a massive single inclusion in it, and just another example there. So these are usually seen, in my experience, in children and pretty rare. This is just to remind me that actinomyces and candida are just normal flora in the oropharynx, and so we don't report those. Commonly seen in bronchial washing specimens especially, and in sputum. This is pneumocystis, and this is a methenamine silver stain. You can see the little dots I'm pointing out inside the round bodies that are about the size of a red blood cell. So these are very hard to find. Again, an opportunistic infection, commonly seen back in the days of the AIDS epidemic in Australia. Very hard to see with a pap stain. They stain negatively. So we used to use commonly a silver stain to highlight them. This is Aspergillus, and it is in a sputum specimen in this case. I'm just showing you here all of the necrotic material that is associated with this. So these are all necrotic dead ghosts of cells, and that can be a bit of a problem if you get, as I'll show you later, squamous atypia with it. So identifying the hyphae is quite important. This case has quite a lot of them, and you can see it's a fairly thick hyphae and stains quite dark with hematoxylin, so it's pretty easy to find usually. There's a lot of it in this specimen. Just on high power you can see the characteristic branching at 45 degrees and the segmentation of the hyphae in this case. And in my experience, cytology is quite sensitive at detecting this. Often we find it when micro haven't on microscopy, and then they confirm it on culture. So we just report this as Aspergillus species. This is an example of some of the sort of squamous atypia you can sometimes get when you get an aspergilloma developing, and they can radiologically mimic SCC and cytologically as well. See the pleomorphic cells here. The key is in noting that they don't have marked nuclear atypia, and obviously identifying the hyphae that go with them. This is Cryptococcus, and this is a pretty easy one. So these are quite variable in size. The capsule, large capsule around the outside of them, often is clear staining. In this case it's staining a kind of bluey grey, and these are quite large compared to red blood cells. Sometimes they're quite small. A lot easier when that capsule stains like that, so sometimes it's quite variable. It'll stain orange with a pap stain. In this case it's this sort of bluey colour. and they're, as you can see, quite large. They have that central round area and then a mucinous capsule around the outside. It's a lot easier to see when there's a background present. That's why India ink is used in micro. But you can see here in this bloody background, it's really highlighting the organisms, even though they have a clear capsule. So you can imagine if there was no background, like a CSF specimen, they're quite hard to see. Even in this sputum specimen, there's sort of some proteinaceous mucinous material in the background that is highlighting the clear mucinous capsule around the outside of the cells. So we can confirm that with, say, a mucicarmine mucin stain. And just here on a slightly higher power, so you can see again nicely that clear capsule. Once you see one organism, they're often all over the slide, and this is what we really want to see. So this one here is showing quite nice budding of the yeast. Just a reminder, so glove powder, which is what this is, can look quite like that. Notice it has an angular outline to it, and no budding. This is a case of hydatid infecting someone's lung, and this is a zoonosis. It's acquired from animals, and the chief host of this cestode, which is a tapeworm, is really a canine, and people get, particularly farm workers, infected with these. They can form large cysts that undergo asexual reproduction and create millions of these protoscolices. So each of these could turn into a cestode. These guys have a little ring of hooks around their mouth, if you like, and they develop suckers around that as well. And these large cysts that need to be surgically removed without spilling any of the little scolices, because they can cause anaphylactic shock and also seed and create new cysts. Often the first sign that you see these are the actual individual little hooklets like this that you can see, and that's a bit of a hint. Anyway, that was a quick tour of infectious agents in lung specimens, and there's really quite a broad range that can be seen and you have to be alert for.